Yes, thank you. So first of all, I'd like to say that I'm very happy to be here today to present the result of my work. My name is Corentin Thuilliez. I'm a master's student and I work at the Gustave Roussy Institute. And so I've worked on a tool that will help deconvolve spatial transcriptomic data, a tool called CellFromSpace. So first of all, let's start with the beginning. First of all, you have spatial transcriptomic. So spatial transcriptomic is an emerging class of technology that will help you analyze transcript in situ. So by being able to detect where the transcripts are located within slices of histological tissues. So there are two main categories of technologies. You have sequencing-based technologies as well as imaging-based technologies. And the first technology I'm going to focus on and explain a little bit to give you some context is the sequencing technologies. So you have the sequencing-based method where the techniques base itself on spots that are located over a slide. You place your samples on top of it, and the RNA will filtrate through your sample and drops into the wells where the elements to construct your libraries are located. It allows you the detections of every single gene within your samples, and the captures qualities and size depends on the technologies. But one of the problems is that, as I'm going to explain here after, is that each of the spots, because of its size, will capture RNA from multiple cells at once. So for instance, one of the technologies that's being used is the Vision technology, one of the most widely spread technology within the labs, with spots that are separated by 100 micrometers each and are 55 micrometers wide. And as you can see here in these pictures, you have multiple cells within each spot that where you're going to detect the transcript from these cells. And so there is a necessity for deconvolution tools for these spots. Most of the technique and the tools that exist base themselves on a single cell reference. But the problem with using a single cell reference to deconvolve spatial data is that, first of all, not everybody has necessarily the means to deconvolve it, or the money for it, or also there are some cells that can be lost with a single cell analysis. And so there is a necessity to develop tools with reference that can allow for reference-free deconvolutions. So that's why we've developed CFS, so a tool for reference-free deconvolution. It's an R package that bases itself on the Seurat type of object. And the way the pipeline works for the tool is very similar to regular single-cell analysis. So from your matrix count, you first have this normalization step, and then you are going to go through an independent component analysis steps, or ICA step. So I'm going to explain what this ICA is and what's the difference between the classical technique called principal component analysis that's being normally used in single cell. So ICA is different from PCA by the fact that it's a technique that will allow you to separate signal. So if you want to understand here, I'm explaining using a very common problem, which is the cocktail party problem, where the voices of two different persons are being recorded by two different mics. So when you end up with two different recordings of the voices with the two different mics. And by using ICA, it's possible to separate the signals of each of the person's voice to have an isolated signal of each of these voices. So the sources here in this example are the person that are being recorded, and the detector are our mic. So if you want a comparison with classic PCA, so you have principal components, which are a way to simplify the observation of your data set. Each of the axes are orthogonal and dependent from one another, meaning that you cannot choose the dimension that you like. You have to keep the entire thing if you want to keep going through your analysis. However, with ICs, independent components, you will be able to immediately and directly detect each of the specific type of signal that you will be able to find within your samples. And also, this signal, these independent components, are independent from one another, meaning that if you ever detect a signal that you're not interested in, such as noise or, for instance, a biological process that you don't want to keep for the remainder of your analysis, such as apoptosis, you can remove it easily in order to not take it into account. So ICA is a method that's already been used for quite some time in EEG data analysis, where you will place multiple electrodes over the cranium of the patient, where each will record a different signal at a different location. And by using ICA, it's possible to detect the signal originated from different locations of the brain. So how are we going to use this independent component analysis on our spatial transcriptomic dataset? So you have this, we consider that the cell types are the source of our signal, where each of the spot transcripts are going to be our detector, where you will find a mixture of the signal of each of the cell within it. And by using ICA, we'll be able to detect each of the specific signal of the cell within our sample. So now that I have explained what ICA is, so as I explained, you can detect cell type, biological structure, and biological function with this technique. And we reach a very important step, which is the IC's annotation. This IC's annotation will be important because that's where you'll decide what the independent component means, whether it's a cell type, a biological process, or something you're not interested in. And in order to make this process easier, we have developed a Shiny tool alongside the R package in order to annotate by hand this independent component. So the way this tool presents itself is by recapitulation of all of the ICs with their associated genes, the spatial localization of each of the ICs. You also have the spatial localization of the genes that are associated with the IC, as well as the list of the IC or the gene associated with them, as well as the enrichment of each of the ICs, of each of the genes associated with the ICs. And all of this will help you annotate and give meanings to those independent components. In the end, you find yourself with an annotated table where you'll have a very wide cell type that you're going to put in it, as well as a more defined annotation where with a very specific meaning of the signal. And with this annotation, you'll be able to proceed to the visualization step, where you'll be able to display, even within the tool, the genes and the IC, obviously, any metadata you might want to display, but as well as clusterize your sample using the independent components. With the tool, you can also display the density of each of the cell types across your samples. And also, you can use scatterpie. You can generate scatterpie plot. So each of the scatterpies represent the IC signal, where the size of the pie charts represent the intensity of the signal overall, and each of the colors represent one of the ICs and the relative quantity between each of these ICs. You can also represent every single of the ICs at once using the scatter pie chart. So what's interesting here now are the results that we obtain with using the CellFromSpace and the ICA method. So here we can see that using the mouse brain tissue with the IC, we'll be able to detect directly specific regions of the brain, such as the cortex layer, but also very sparse tissues, cell signal, such as microglia. And by clustering, you are able to regenerate the clustering, the organization of the brain tissue with a relatively high fidelity depending on the resolution of the technique that you're using, of course. But also, it doesn't just work on brain tissue, it also works on very complex and disorganized tissues, such as here it's an example with breast cancer, where you can find the annotation that's made by a pathologist. And the clustering is able to recapitulate kind of the organization that you have, that the pathologist has given to us. But also one thing that I want to insist on is the fact that for spatial analysis, using clusterization isn't necessarily the most pertinent method, especially for complex tissues such as cancer tissues, because you have mixture of cells within each of the spots. So what we do is that we remain within the latent spaces. So as we can see here, by just observing the ICs within our sample, we are able to recapitulate each of the signals found within our sample as well as their localization. You can already kind of get an idea of the organization of the tissue with just it. But with the annotation, you're also able to observe the localization of the cell types as well as their relative quantities between one another. In addition, the tool can also allow to display the density of specific cell types, or also allow the subsampling of your samples. Here, for instance, with subsample based on the spot that contain signal of cancer cells. And with this, you are able to create, to re-roll the pipeline in order to create new cluster and see how the cell evolve across your tissue. So we've not just used this package with the Visium technology, we've also tried to use it with a SlideSec technology, which in terms of analysis and output is very similar to Visium, except at a much higher resolution of 10 micrometers. And as we can see, even if you don't necessarily need to deconvolve a signal, IC can also be used to find to detect a very accurate representation of your tissue here in the mouse hippocampus compared to the reference which you can see on the right here. Now I'm going to explain probe-based technology, because we've also tried to use our package with these type of technologies. So these type of techniques are very different from what I've explained earlier. Instead of using spots here, you're going to use probes with a fluorescent signal in order to detect specific numbers of transcript from between 500 to 1,000 genes. And because this technique bases itself on imagery, you get a very precise, very, very high resolution on the data that you obtain in the end. One of the challenges with this technology is the cell segmentation, where there are still challenges in order to get an accurate enough segmentation of the cells with these technologies. One of the problems also is because the results that you obtain at the end is a matrix containing each of your transcripts within your sample, the results are very hard to manipulate because of how massive they are. And so here I want to show you that the package can be used to detect region of interest with these technologies as a first pass analysis. So the way this works is that we create pseudospot by creating a grid over our sample where we'll place each of the transcript at the appropriate location, and then by rolling the pipeline normally, you end up with something that's very similar to Visium. And so we've used it, for instance, here with the MERSCOP mouse brain receptor map with a 40 micrometer binning. And we were able to find that 254 genes contributed to these ICs. And as we can see here, we are able, with clustering, to reconstitute the organization of the mouse brain tissue too. Just like Visium, we are able to detect sparse cells signal, as well as a specific mouse brain area. We've also used the package with the Cosmic technologies. One of the advantages that we had with this technology is the fact that we have multiple samples of lung cancer tissues, which we've used along with a ground-based truth. And as we can see here, compared to the base ground truth here, we are able to, with clusterization, to reconstitute the organization of the tissue with relatively high fidelity. And also, we were able to start checking if the IC that we were observed and the intensity of the signal really correlated with the base truth of the B cell count. So here you have two different ICs that were annotated as being B cells of different types. And as you can see here, you have you're able to really see the signals of that really correlate with the B cell counts using the ICs. Here, there is the same thing with tumor cells. So tumor cells are very heterogeneous. So there are a lot of different ICs. Here, I'm only showing one, so you don't have the entirety of the signal of the tumor. Because we also have multiple lung samples, we are also able to try an integration using independent component analysis. And we were able to show that this method can extract directly within the tissues the signal of cell type that are in common between all of our lung samples, as well as the signal that's specific from each sample, such as the cancer cells that are very different depending on your samples. And we end up with something like this, where the tumor samples, here we have in red the cells that are in common between each of the samples. And here we have what stands out from the rest are these tumor cells that are very heterogeneous. So they are separated here on our UMAP. Here I'm showing you some specific signal from our integrated data set using the references. Here for the tumor from the sample 5. Here from the tumor from the sample lung 9. The B cells across all samples. And here you have the monocytes, which are located in an area where you'll find all of the tumors, because they are mostly found on the spots where are located tumors from the lung 13. So in the end, we're using RCA CFS can deconvolve with no prior knowledge the cell identities, biological features, and morphological structures based on expression patterns. We work by remaining in the latent space of the independent component. You can subsample for final analysis. It works with all currently available technologies, and you can easily integrate sample. So if you're interested, there's a beta version that's still under works, available on the GitHub if you want to try it as it currently is. Here are my contacts, and a preprint will soon be available. And I also want to thank all of my team, especially Antonin Marché, which is my supervisor. And now I'm ready for your questions.
Due to time, maybe we can have the next speaker come and set up while we take any more any questions for our speaker. Do we have any questions? Yeah.
With independent components analysis, the components aren't ranked in any way, and you need to identify the number of components that are relevant and the number of components that are maybe spurious. How are you doing that?
So I'm going to place myself closer. So so far we haven't. So we kind of left that up to the users. We haven't implemented a method to choose an ideal number of independent components, because it just works even if you don't try to necessarily try to find an ideal number of independent components. For instance, for analysis, we usually use 100 components. And even if there is some redundancy, usually you find what you find are subtypes of different cells or different cell types. So it's not really a problem. You just annotated as it should be.
But there's no reason why, like, the most prominent component wouldn't be number 256 or number 1, just because of the way it works.
Maybe we can talk about this, or do you
Yes.
have an answer?
Not right away, so
Okay, so maybe you guys can discuss it offline.